We will wait a little bit for the participants entering, okay?
Okay.
So we'll start. So greetings, everyone. Welcome to our first session of the EMN webinar series of this year. My name is Diana Pinto, and I'm a final year PhD student at REQUIMTE LAQV, Polytechnic of Porto from Portugal, and I am co-hosting this webinar with Domenica Berardi from Yale University, USA. On behalf of the EMN from Metabolomics Society, I would like to thank you for joining this webinar, and I hope you all enjoy it. So also I would like to encourage you to follow the EMN and Metabolomics Society online on Twitter and LinkedIn, where we share some exciting news about the committee, the MedSoc conference, travel bursary announcements, and some other interesting opportunities. So please connect with us and follow us online. Also, we make here available the EMN email in case you would like to contact us by email. And today I am very happy to introduce Professor Dr. Rosa Lamuela Raventós and Dr. Julian Lozano Castellón from the University of Barcelona in Spain, who will talk us about the Polyphenol Research Group, Food and Nutrition. It is my pleasure to introduce Professor Dr. Rosa Lamuela Raventós as our first speaker. Professor Rosa is a full professor at the Department of Nutrition, Food Sciences and Gastronomy from the School of Pharmacy and Food Sciences, University of Barcelona. She has been one of the most influential scientists in the world, according to the highly cited researchers list published by Clarivate Analytics during 5 consecutive years. Since 2017. Professor Rosa is also the leader of the Polyphenol Research Group, which has been recognized as a consolidated group by the science ministries of the government of Catalonia, and she also leads the CyberOBN Research Group since 2012, and was the director of the Research Institute of Nutrition and Food Safety since 2015 until 2023. Her work as a researcher and professor was recognized multiple times with national and international awards, and Dr. Rosa is also involved in national and international projects related to nutritional interest of bioactive compounds present in foods and natural products and their impact on health. Indeed, I was privileged to be a visiting PhD student at Professor Rosa Lab, so I am very glad to introduce her. And please, Rosa, the floor is yours.
Thank you. Well, I would like to thank you to Diana and Domenica for having invited us. Diana was in our group. It was really a pleasure to have her in our group. Her productivity is incredible. So we are really pleased to have you whenever you want. So I will try to put my presentation. Okay. Is everybody seeing it? Is that fine?
Yes, but at least I can see the not only the slides, but everything.
So one moment. I will have to...
Presenter mode, yeah.
Probably will have to, what I will do is to do that. Oh, no, but then,
No.
no. Yeah,
In-
I know what I will do. Bye-bye. Now I think it will work. Now it's working?
Yeah, how's it? But you need to put it in a full mode, I think, in the
But I think I have it in full mode. Okay.
like in
Espera, one moment. Now?
No.
No?
Rosa,
Sí.
guárdala como a PDF.
Pero si ahora lo vais a guardar, espera.
Bueno,
Sí, espera, compartir. Compartir. No, no. Bueno. Pues no, pues farem. Vale, espera. We'll do that. Sorry about that. But this is not the we will see. Now I think it should work. Are you seeing it or
no, just the whole.
no?
it is
I
not
think
in-
it's fine. We can go on like this. It's okay.
It's
Yeah.
okay.
I mean, Rosa, I can share the screen. I have the and
The
I can
presentation.
try it.
Okay.
Yeah.
Yeah, that's fine.
So yeah, if you stop. Oh well, I think.
I stopped sharing, no?
Yeah.
Sorry. Espera.
See. So now, do you see it?
yes, it's in full mode now. Yeah.
Okay.
Okay.
Well, sorry about that.
No problem. Okay.
Well, today we will talk about metabolomics offering an avenue for studying dietary patterns through biomarkers. And this work has been done in collaboration with several members of our research group. But before starting with the talk, I would like to introduce the group. The group nowadays, the polyphenol research groups, it's led by myself, but we are 3 professors and also 3 postdocs, 1 Marie Curie and 2 postdocs that are from Spain, and 1 project manager. Moreover, we have 8 students, 8 PhD students that, well, we are very proud of them because we have students from all around the world, from Colombia, Egypt, Russia, Chile, Paraguay, Mexico, because we are open to collaborations and we are happy that people from all the world comes to our research. You want to pass the next slide? Moreover, we are also collaborating. For example, Ana Verdu, that is one of the professors of our group, is collaborating with another, leading the work with Dr. Pinto, the PhD thesis that will be defended very soon from the Ana Pinto. And moreover, Ana and Maria, that are also 2 professors from our research group, are also involved in 2 PhD directions. And this is very good, I think, because then we do intercollaboration with other groups. Next slide. Next slide. Yeah. It's okay. Well, I have divided the presentation. The presentation will be focused mainly in polyphenols. And we will talk initially about polyphenols intake, but what happens when we measure polyphenols intakes? Then we will go to studies that we have performed in plasma versus urine, which is the best matrix to use or the best conditions for analyzing polyphenols. We will compare analysis that we have performed targeted versus untargeted, mainly in urine. And finally, Well, we will do a take-home messages where we will talk about other human samples to explore. Go ahead, please. Well, when we talk about polyphenols, and these are the compounds that are most abundant in our daily diet, the problem that we have, well, problem or advantage we can say, is that they are very spread in nature. We have in many plant-derived food, like fast fruits, vegetables, cereals, legumes, and beverages, coffee, wine, tea, etc. Moreover, what we probably is more complex about polyphenols is that here there are many compounds inside polyphenols from different families, and more than also different families, they are bound to different acids, to different sugars. So there are many structures. It says, but I think there are many also to be discovered, that there are more than 9,000 polyphenols in our diet that we can consume. Next slide, please. Yes, initially we started to analyzing what was our polyphenol intake, and for that we use a very famous database that is Phenol Explorer. We did that in collaboration with University of Harvard, one of that now is professor in our department. One of our, by then, PhD students went to Harvard, and we analyzed what was the polyphenol consumption in the PREDIMED trial. That PREDIMED trial is a trial that has been performed in Spain in elderly population. And what we observed is that the polyphenols consumption in average was around 800 milligrams per day, more or less. However, what we observed that was different from other populations is that wine is very important in the consumption of polyphenols in our population and this elderly population. And also one thing that was different, especially if we compare people from the US or people from the north of Europe, is that olive oils and oil was also important in the polyphenols intakes in our population. After that, what we did is we were lucky to participate in the PREDIMED and have a lot of data. We had food frequency questionnaires, we have urines, we have blood samples, and what we had also were health events, that we could measure what was the effect of the polyphenol intake in health events. And for that, what we did is a longitudinal analysis measuring polyphenols intake, accumulative polyphenols intakes during the years, and what could be the effect of this polyphenol intake in health events. Go ahead. And initially we measure in cardiovascular disease events. That was the main aim in the PREDIMED study. The cardiovascular events were, for example, stroke, myocardial infarction, mortality for cardiovascular events. And after we did it in mortality, what we could observe in both is that total polyphenols has a significant impact decreasing cardiovascular events. Moreover, there were specific polyphenols that were having also a protective effects like lignans, isoflavones. And in mortality, we observe also the same, that polyphenols had a protection effect. Moreover, there were some of these polyphenols that has an effect. And if we look in which foods these polyphenols are present in our population. In our population, for example, flavanols were coming mainly from cocoa and red wine. Hydroxybenzoics in our population were coming mainly from red wine also. Lignans in our population, usually they came from whole grains, but in our population were coming especially for virgin and extra virgin olive oil. Stilbens, well, usually they came from red wine or grapes. And in the case of isoflavones, in our population is not usually to have soy products, especially in the population that is over 50 or 60. Usually they don't have many soy derivatives. So they were coming mainly from beans. However, if we go to the next slide, however, polyphenols, we know that they have many health effects, not only related to cardiovascular disease, also to the neurodegenerative, cancers, obesity. So we were interested in measuring them also in biological samples. Next slide, please. Yes. What is, well, there are some advantages and disadvantages not to use polyphenols. It's good to have like a biomarker of polyphenols intakes, however, because then we can measure what you are consuming, because there are, well, as you have commented, no, there is a diversity of polyphenols. Sometimes it's difficult to know exactly what is the intake, only considering the food frequency questionnaires. Moreover, with the food frequency questionnaires, we are not considering that there is different variability and there is a diversity on the metabolism. No? Some polyphenols are more metabolized than others. So the strength of having biomarkers is that they are more objective, they are more accurate. However, we have some limitations, a lot of limitations. Difficult sometimes to select the best biomarker. Sometimes it's difficult to perform these studies in large-scale populations. They are much more expensive than the polyphenols intake. And sometimes we have to very complex methodologies to analyze these polyphenols. Next slide. Okay. Well, and then which samples do we use? This is when, for example, we collaborate with epidemiologists, they always ask, do you analyze blood samples? Because usually it's what is more common in the people who have performed big studies, no? To have blood samples. Or we will see what is good of having plasma or blood samples versus urine. Well, this is a pharmacokinetic of any compounds, no? There is this absorption curve, no? That you have a maximum absorption and then you decrease. But if we look at what happens of polyphenols, if we look at the next slide, what happens with polyphenols? That that in the polyphenols, here we compare, for example, the case of resveratrol. This was a red wine, grape juice, and red wine extract with the same amount of resveratrol. If we look, usually, at 24 hours, there are not almost any residue of polyphenols, of resveratrol in that case, in blood. So it's difficult to measure them because they have usually short viability, especially the ones that are present in food, no? However, in this case, I would like to mention that in the wine, if you see the viability was higher than in the juice, giving the same amount of resveratrol, no? And in the juice, the viability of resveratrol was higher than in the pills. Then we performed studies about resveratrol, about polyphenols viability, considering what was the effect of processing a matrix on the viability of polyphenols. For that, we have performed different studies, intervention trials. In that case, we performed an intervention trial in 4 healthy population. Well, this was a prospective, randomized, crossover, open control design. And here, the participants on this study had to take the 500 grams of tomato or the same amount of tomato as sauce. So you have to consider that some of the water has been eliminated, but this would be the same amount and the same tomato. They were prepared by our professors of technology who prepared this sauce. And in the sauces, in another item, we prepare also sauce, but adding olive oil. So we analyze urines and samples. What we observe in the case of samples, in the case of plasma, we observe that, as we observed previously, that at 24 hours there is no Talmud curves. However, a curious thing is that when we add olive oil, there is not only one peak, there is like 2 peaks, and this was observed in different polyphenols that were present in tomato when we analyzed them in blood samples. No? There was a double peak, so it was like an absorption, then a decrease, and then again an absorption. Next slide, please. So what we attribute, what could be this, why this is happening, no? We attribute that to the enteropathic circulation, no? That when you have this compound, then this compound is absorbed, is eliminated to the intestine, and then is reabsorbed. For this reason, we have this double peak absorption. That this is interesting because in one way you are increasing the viability of these compounds. This could be in blood. Next slide. And in blood, we have, well, this happens a lot in our intervention studies, that we observe very big difference between men and women. We have very big difference according to the gender. Moreover, this is not only, it happens to us, it happens to almost all the studies, that there is a big standard deviation. And when we look in urines, we can see here will be tomatoes, and here will be tomato sauce without oil and tomato sauce with oil. What we observed is that in general, when you process the tomato, there is an increase in these compounds in urine. So the viability of these compounds increase, and they usually stay more time in the body, and this means, I am not presenting the results, we look at the anti-inflammatory effect, that they have more anti-inflammatory effect because they remain more time and they are more bioavailable in the body. Next slide. And here is also giving tomato during 24 hours. In this case, we measure total polyphenols in urine, and here is what will happen when you measure polyphenols during 24 hours urine excretion. You see that there is an accumulation of polyphenols when there is this when you take polyphenols during 24 hours, no? There is an increase on these polyphenols during the 24 hours. So polyphenols in urine, the good thing that we have is that we have them more time in the body, and we can analyze more easily. Next slide, please. So what you can conclude from that, no? In plasma, we have much less amount of these compounds because they are diluted in 5 liters of blood. Moreover, they have shorter time because like they are more hydrosoluble, they are not less vitamin D or vitamin A, that they are long time in the body. And so they are more difficult to detect them. In urine, we have more concentration of these metabolites and they remain through longer times. Next slide. Well, and we'll see the difference that we have observed in some of our studies using targeted versus untargeted in urine. No? As you know, if you look at the next slide, no, when we go through target analysis, the good thing is that you are more focused and you are looking specifically of what you are trying to look. No? For example, of one polyphenol that you try to look in urine. When you are doing metabolomics, it's like you don't know exactly which will be the compounds that will give the difference. No? However, you can have much more information. And this is a very interesting thing of using metabolomics. Next slide. Well, and here are some of the results that we have obtained. This is a study that we performed in an adolescent population. These adolescents have been followed up during 4 years. However, I will present the results in the baseline. We were also very lucky to participate in this project, collaboration with one famous cardiologist, Dr. Valentin Fusté, that the main aim is to try to improve the health of this population. But however, talking about phenolics in urine, what we did, well, we performed like a solid phase extraction to clean up the samples, and after we analyzed the sample through Orbitrap MS. Go ahead, next slide. Well, and we could detect 50 polyphenols. However, what was good is that some of these polyphenols, and this has been observed also in other studies, some of these polyphenols were very well correlated, so can be used as biomarkers. For example, from nuts, urolithins, and from cherries, they were correlated with nuts and cherries. And for example, in the case of stilbenes, this is the dehydroresveratrol that is coming from the microbiota sulfate. This is a compound coming from the microbiota. We could relate it with great consumptions. Go ahead. Moreover, there were also other compounds as enterodiols or enterolactones that are coming also from microbiota that we could relate them or we could associate them with higher fiber consumption cereals, legumes, seeds, and the consumption of fruit and vegetables. Go ahead. Moreover, we have performed also the same analysis in other populations. In this case, also we perform in the Predimed population, as we have explained it before. In this case, we perform it in 200 populations, and we did also the same procedure. And what we observed that the polyphenols, microbial phenolic polyphenols that were coming from microbiota, we could associate with higher adherence to a Mediterranean diet, a questionnaire that is done to the population to see if they are following a Mediterranean diet. Moreover, we could relate it, that is the most interesting part, I think, to a better cardiovascular health, so low or lower risk cardiovascular risk factors. Next slide. And here, I would like to compare what we obtained from using targeted versus untargeted. In this case, we analyze one specific polyphenol from beer that is isoxanthomol. Isoxanthomol is coming from the beer, is from the part of the lupus of the beer and during the brewing process, this compound is xanthomol is transformed to isoxanthomol. And we could detect these compounds in urine, and they were, as you can see in the next slide, we were very good biomarkers from urine. For that, what we did is a dose-response randomized crossover clinical trial in 20 men and 20 women. They were young volunteers. And what we did is they consumed different amount, a dose response. This was 0, 1 beer, 2 beers, and 3 beers. And in the case of women, like they can have less amount of alcohol, they were 1 beer, 1 beer and a half, or 2 beers, no? The night before doing the study. And what we observe in that case, using the isoxanthomol as biomarker, we see a clear dose response, no? That if you analyze this compound in urine, especially males, there is a clear dose response according to the amount of beer they have consumed. However, we observed also a gender difference. No? In the case of women, there is a difference between 1 and 2 beers. However, here there was 2 different responders, one female responder that has lower excretion, and one having the same amount of beer, and another females having higher excretion. So there is different response, and we did not observe this response or this behavior in the case of men. Okay, please. Well, and after we also did this study to corroborate if it was really a good, reliable biomarker in a clinical intervention trial that we use beer without ethanol, free ethanol beer, and gin. Gin was the control, no? To have ethanol, but not to have the compounds that are coming from the fermentation. And here is the results. After the washout, we cannot see isoxanthomol at all. In the case of gin, we cannot see isoxanthomol. However, when you see beer, after the beer consumption or after the beer without ethanol, we see that there is a clear response. And we did a rough curve, and we have very good result. No, go ahead. So we can conclude that, but what we did also to really verify that this is a good biomarker, that this to have a good biomarker is that you don't know really what the population has consumed, and analyzing their samples, in that case urine samples, you could detect you have drink beer, no? But they can be lying, no? But you can detect if they have been drinking beer or not. And what we did is in the Predimed population, we take through the food frequency questionnaire people who were not consumed beer and people who consumed beer. And we could see clearly that we could detect perfectly the people who had beer or not. So we can say that isoxanthomol were really a very good biomarker that can be used in epidemiological studies to differentiate between beer and not beer. But after that, we decided to do an untarget analysis, an untarget metabolomic analysis in the same study that we have described before, using beer without ethanol, and gin. And if we look at the results, if we go to the next slide. Yes, here, if you see the results, we can, well, there are different results. Here we can see, for example, the 4 classes. Here we see beer and baseline, gin and baseline, and here there is beer with ethanol and beer without ethanol. But if we go to the next slide, in the next one, you can see clearly. Here we can see clearly which are the compounds. There is one, if there is more movement in this slide, if you can pass, please, Julian, you will see that, but you can do it directly. Yes. So if you can see, we could see this was the good thing of doing untarget analysis, that we could detect compounds that were clearly coming from the alcohol metabolism, in the case of the gin and beer with ethanol, compounds that were coming from hops, from beer, that were present in the beer and in the beer with ethanol and beer without ethanol, and compounds that could differentiate beer that has come from the fermentation of beer, that they were compounds coming from the fermentation. But moreover, what was more interesting for us is that we could detect that when you had beer, and we could not detect that when after the washout or after the gin, that there was some compounds that were in higher concentration that differentiated as the compounds, and they were coming from that there was an activation of the mitochondrial metabolism. So this is very interesting because you can have much more information than when we use target metabolomic analysis. Next slide. So what we can conclude from this result, that using target analysis, we could elucidate a new biomarker, in this case isoxanthomol. The isoxanthomol were really very specific of beer, so it allows to discriminate beer as non-beer consumers, and we can use it in larger studies. However, when you use untarget analysis, isoxanthomol could not be detected. So different compounds can be analyzed or can be described in different in the 2 ways of analysis. However, other compounds discriminate among beer and non-beer consumption. Moreover, endogenous metabolites were also identified, that can give an idea of what is happening in the human body. The next slide. Well, and this is another analysis that we have also performed, that this is very recent analysis in the PREDIMED, and doing this was done by Ines, and what we observed is that there was a clear association of microbial metabolites with diabetes too, and we could differentiate the individuals if they were having diabetes or if they were not diabetic. Here you can see clearly the difference between the 2 groups. We go to the next slide. And the compounds in that case that differentiate or that allow us to differentiate between these 2 groups, the diabetics and not diabetics, were these 2 compounds, one that is a phenolic acid and the other is a flavonoid. Next slide. So what could be the take-home messages? So urine samples can allow us to measure polyphenols intakes. In blood samples, it's much more difficult to measure polyphenols intakes. There are some polyphenols that can be measured, but in very low amount, and usually the ones that are coming from the microbiota. Target and untarget analysis are very useful tools that give very different results and can be complementary. However, I think we have to explore still other samples. No, for example, we have got very good results using feces samples that I think they need to be much more explored. And for example, saliva samples. No, there are samples that are easily to take that can be also easily explored. And finally, well, I would like to thanks to all of you for allowing us to participate in this interesting seminar. And if you want to follow up in media, we try to be very active also in media, trying to show the results that we are obtaining or the activity of our research groups. So thank you very much. And if you have any questions, I would be very happy to try to answer. Julián, deixar de compartir.
Thank you, Rosa. So I think we have one question. So one of our participants is asking how polyphenols are absorbed by the human intestine. How are they biotransformed by human enzymes and the human gut microbiota? Thank you.
Yes, well, usually they are absorbed through a specific mechanisms, through like we have a specific mechanism to absorb polyphenols, especially the ones that are hydrosoluble. And the thing is that in the enterocyte, they are transformed to glucuronides and to sulfates. So usually you don't detect the same compound that was present in food. It's easily transformed in the enterocyte, and this transformation follows in the liver. No? So they are really, we have a lot of transformation of these compounds, like they are considered xenobiotics, just they enter in the body, they are quickly transformed. And a question about the microbiota, usually the polyphenols nowadays are considered prebiotic substance. So they are like food for the microbiota, and microbiota polyphenols are good for the microbiota, they grow, and microbiota transforms it to other metabolites. Usually they are transformed to more smaller molecules that are also more easily absorbed, and they are absorbed through the colon. No, these are the ones that are coming from the microbiota, but then they suffer also transformation. They also suffer sulfatation, glucuronidations that are finally the ones that we can detect in urine.
Thank you, Rosa.
Another question I got from the Q&A is about the process for polyphenol annotation in untargeted method. Like what is the process for polyphenol annotation in untargeted method?
Sorry, is it the annotation?
Yeah, I can answer to this question if you want, Rosa, because
Yeah,
it's
for sure.
mainly my
Yes.
work. And well, we actually use the MS Dial and MS Finder, as Diana knows now, but then we check manually each annotation that the program gives us, because I mean the MS Finder is very useful, but we have problems with the isomers because it just checks for the molecule. But if you have a P and an O, then you have to check. For example, in the case of the Kumarics, it always go ortho meta para, so it's meta para and then ortho in the reverse phase chromatography. So like this, you can check which compound is each one, and then we have also a pool of the standards. So we use them to the annotation and to differentiate between the isomers. That's mainly we mainly use the MS Dial and then the MS Finder, and then we manually check everything.
Thank you, Julian. One more question. So why are the concentrations so much higher in wine than in juice or extract? Rosa?
Oh, yeah. In blood or because they are, well, I don't know if the question is if they are higher in the food or in the blood.
I think
No,
in the food.
in
Yeah,
the blood that you presented the results.
Okay, in the blood.
No.
Yes, because well, we think that in the case of the wine, there is ethanol, that ethanol improves the viability of the trans resveratrol. And in the case of the juice, it's higher than in a pill, because usually it's more bioavailable. It's the same that it happens with us with tomato, no? If you have the same amount of polyphenols present in tomato or tomato juice are more bioavailable than in tomato. Because the same happens with a pill. That is like a solid part, and usually as a solid form is less bioavailable than when you have in a liquid matrix.
Okay, thank you, Jose. I also have one question.
Yeah.
It's more general. It's what do you think? What do you think they are the main challenges on identifying polyphenols biomarkers regarding the metabolomics analysis?
Well, I think here we have several problems. Well, one is that there is the standard deviation in humans are very high. So this is a big problem that we have with analyzing polyphenols in humans. And having the same amount of polyphenols, there is a high standard deviation. This is a problem. However, using it as a biomarker, the main problem we have is that a lot of polyphenols are not specific from one compound, no? From one food. So to have a food that has a polyphenols that is specific for this food, sometimes it's very difficult, no? Because they are present. Quercetin, for example, no, is present in onions, in samples, in wine. So it's very difficult to find the perfect biomarker. However, the good thing now is that in general, in epidemiology or in health, it's more interesting the dietary pattern than the food. So now we are more interested in to look for a diet polyphenols that are biomarkers of a healthy dietary pattern. Like, for example, we observe with the people who were having a Mediterranean diet that they have more extraction of polyphenols. And I think this would be the good thing, no? That if you are following a Mediterranean or a pattern, it will be more easy to see if the people are having, for example, a diet rich in fruit and vegetables, or a planetary diet, or are having like a ultra-processed food diet, no? This, I think, would be the most interesting part.
Okay, thank you, Jose. Thank you so much. Great talk.
Thank you.
I will give the floors now to Domenica for presenting our second speaker.
Juliana. So for other questions, I'm pretty sure that Professor Lamuela Ventos will be more than keen to answer the questions also by through emails if you have more.
Yeah, for
and
sure.
Thank you so much for the great presentation. So it is my pleasure now to introduce Julian Lozano. Apologies for the pronunciation of the name.
It's okay.
Julian is a postdoctoral researcher at the University of Barcelona. His research focused on targeted and non-targeted analysis of multivariate statistics to find relationships that allow a better understanding of interaction between food and health. He did his thesis studying the effect of processing on extra virgin olive oil metabolome, focusing on the effect of cooking, and he also did several national and international collaborations, mainly focused on the reuse of agrological subproducts and the effect of different food and extracts on health. Moreover, according to Scopius, he has 26 publications and an H index of 12, and several publications as first authors. So it is my very pleasure to introduce to you Dr. Julian Lozano Castellón.
Thank you very much. And first of all, thank you for the invitation and having this opportunity talking here. And now I'm going to start with my presentation. I will share my screen. Yes. Do you see my screen?
Yes, we can see it.
Okay,
Yeah.
and now,
In full mode, yes. Thank you.
yeah, in full mode. Okay,
Yeah.
so now I'm gonna talk about the targeted foodomics analysis that we have just recently published this winter, or no, in autumn, at the end of autumn, and that it was focused on the impact on Evo composition of processing, ripening index, and storage. And the presentation it's divided in those sections. I'm going to talk about extra virgin olive oil, then our samples, the targeted analysis we carried out, then the statistics, and then the results. First of all, extra virgin olive oil is just the fatty fraction of olive juice. It undergoes no refinement process. It's just mechanical extraction and then decantation or separation. And by the regulation, actually by the International Olive Council regulation, it should have no organoleptic defects, as light fruity taste, and the acidity shouldn't be higher than 0.8%. Evo composition is divided in two. One saponifiable fraction that has mainly triglycerides, diglyceride and monoglycerides, and also fatty acids. And the main fatty acid of these triglycerides and all these glyceride chains is the oleic acid, which is a monounsaturated fatty acid. And then it has the minor fraction. This minor fraction could remain in this oil because there's no refinement process. So there's no elimination of any compounds. In this minor fraction, we found several bioactive compounds which gives this importance, this health importance to this oil. And we found the phenolic compounds, also the pigments that are we have the carotenoids, mainly lutein and also the chlorophyll. Also, we have tocopherols, esqualene. We have volatile compounds too, and also the terpenic acids. In this case, I just put it here, the oleanolic acid. And this extra virgin olive oil is done as follows. First, there's the harvest, the picking of the olives. Then there's a cleaning of the olives because normally they are mixed with the leaves. So by blowing the leaves or separating by different density using water, they separate the olives from the leaves. Then there's the crushing process. Also the malaxation, which the malaxation is just mixing all together the crushed residue. So you cut the oil drops that are really small inside this paste. They can aggregate during this process, this malaxation process, this mixing process. They can aggregate. So then you finally have big drops of oil. So then you can separate during the decantation from the solids and the water. And then the filtration to eliminate any remaining solids inside, and finally the bottling and the storage. All this process is very important because each step of this process modifies this extra virgin olive oil composition and then could modify its taste and also its health benefits. The mainly factors that have been studied and that we study also in here are some agronomical factors. For example, the irrigation. If there's lots of irrigation, then there's a lot of water, and this oil, as during the extraction, will be separated with a lot of water. Some compounds will migrate to the water phase instead of remaining in the oil. So you can lose some phenolic compounds if there's a lot of irrigation. Also, the latitude and the altitude, the impacts on the weather, so it will impact on the final oil. The orientation of the plants, if there's a part of the mountain that is in the south, will receive less light, or more light if it's in the south, and less light in the north. So this also have an impact. And also the harvest date is important because the ripening index, the oil done with ripe or unripe olives, it's different too. And also the technological factors that are the ones that could be more modified during the crushing and the malaxation process. There's effect of temperature, but also there's effect of the inside enzymes of the olives. So this modifies the composition of the olive oil. Also, if there's the stoning previously to the process of the crushing, then you don't have all these enzymes coming from inside the stone. So there will be less modification during the extraction process. Also, the decantation, using different types of decantation could result in different olive oil composition. And finally, the bottling and the storage could influence. It's not the same to bottle in tin cans than to bottle this oil in plastic bottles. And the storage, it's not the same to storage it freezing or just in like room temperature. So this is what we wanted to study, mainly the different factors that were affecting this extra virgin olive oil composition and how these factors affect it. For this, we developed the following study with those samples. We wanted to study the different ripening index, the different malaxation time and different malaxation temperature. And also we did a 9-month storage in order to know how this storage affects this composition, how this during this storage the oil is degraded. We selected 9 months instead of 1 year because there's normally different varieties of extra virgin olive oil that the harvest is on different dates, and also there's the early harvest, the late harvest. So at the end, there's harvest from olives from October to December. So you have a period of 9 months that you are consuming olive oil until the starting of the next harvest. So we decided to do just the 9 months storage. And also we studied the different, like, let's say, usual times and temperatures for olive oil extraction. And also the ripening index, we were using the, let's say, green olives because it's more usual to do olive oil, or at least let's say a stable olive oil with green olives, that it's known to have a higher concentration in antioxidants, in phenolic compounds and vitamin E. And well, first of all, yeah, I will explain it now and not later. The ripening index is measured by color. It's just the color of the flesh of the oil, of the olives, sorry, and the color of the skin of the olives. And then with these samples, after having done this oil in a lab machine, in a lab scale machine, we did the storage simulation using plastic bottles. We decided to use plastic bottles because they are not so good to maintain the extra virgin olive oil composition. They are like worse, so we could see more changes in this 9-month time. And also we protected these olives from the light, but we left them in room temperature without any control, just monitoring this temperature, but no control of this temperature. And with these samples, then we did all targeted analysis, but several targeted analysis. We firstly did the quality parameters in order to see if this oil is extra virgin or another kind of oil, like virgin olive oil, not extra virgin olive oil. Then we did the fatty acid profile to know the fatty acids inside. And then the minor compound study, we did the phenolic profile, the volatile profile, the sensory profile too, which was done by the official panel test, and they were testing for 49 sensory attributes. And then also we studied the esqualene and the vitamin E. And well, now let's make a brief parenthesis. We decided to do a targeted analysis for different reasons, because I mean, in the targeted analysis, you have the identification of the compounds is already done, because you normally use the sample. I mean, well, previously to the target analysis, you have to identify those compounds, but when you have identified those compounds, you don't need to do the identification each time. So, and we already had these methods validated, so we could calculate the accurate concentration with them, and we had this equipment for doing those analyses. And we did some of these analysis by ourselves, but some of these analyses were done in Italy, in the case of in collaboration with the University of Bologna, for example, in the case of the volatile compounds. And the sensory analysis was done in the Institute of Research and Technology and Agrotechnology of Catalonia, which they have the official panel test. And in the case of the target analysis, on the other hand, even we selected this kind of analysis, those are really time consuming, those are reagent consuming, and as Rosa said, there's lots of information, because as in the case of Rosa was explaining, here we're just doing some compounds, but maybe there are some compounds that we are missing and could be important. But as we have all the equipments, we were doing this. And after those analyses, we did the multivariate statistics and in order to reduce the important variables and only focus on those variables that really are important in order to discriminate between the different storage times, or in order to discriminate between, I don't know, 30 and 45 minutes of malaxation. And we did supervised models to see, to do a descriptive analysis, and to help us how to understand better the nature of the samples, how those group naturally, and then in order to select better these supervised models, we actually did the PCA, and then we did depending on the factor we were studying, we did a PLSDA for classifying or an OPLSDA for classifying. And then in the case of the marker compounds, we selected those with a VIP score higher than 1.5, those that had a false discovery rate in the case of the student or the ANOVA lower than 0.05. And yes, sorry, this in the case of the now we're gonna see the results, and this is gonna be better it's gonna be more understandable. We did a PLSDA in the case of the storage time, ripening index, and malaxation temperature, and then we did an OPLSDA in the case of the malaxation temperature. The OPLSDA, it's like a PLSDA, but also has inside the model does a orthogonal projection, which inside the model knows the difference inside each group. So it's like doing a SIMCA and then a PLSDA. And it helps, actually, it's for helping separating those samples that cannot be, those groups that cannot be separated with a PLSDA. And also we did an ANOVA at the student depending on the levels of the factor. If it had 3 levels, we did an ANOVA. If it had 2 levels, like in the case of the ripening index, we did at the student. And then here you can see the PCA as the fresh extra virgin olive oil, which is on the right, is really separated from the rest. And there's a tendency on this PC1 that is the storage time. And this PC1, it corresponds to the 26% of the variability. And on the left, we have the 9 months, in the middle, the 3 and 6 months, and on the right, the 0 months. Then if we color differently, this PCA, it's exactly the same, but there's different coloring. We can see that the PC2 corresponds to the ripening index. In this case, we have already colored by a dichotomical factor, low and medium, but this ripening index is a continuous variable. Actually, it goes from 0 to 7, but in our olives, it goes from 1.16 to 2.20, I guess, if I'm not wrong now. And what we saw in this PCA, it was that it was creating two groups with those olives before 1.45, sorry, here colored in green, separated from those olives, from those oils coming from olives with a higher ripening index than the 1.44. So we decided to create this dichotomic variable instead of the continuous variable in order to make it easier for the following statistics. And then just comparing two groups, not having a continuous variable. Then taking a look to all the PCs, the principal components of the PCA, we could see that the PC4 was corresponding to the storage. No, sorry, here it says colored according to storage time, but it should be saying colored according to malaxation temperature, which were 20 to 25 and 30 degrees. And in this case, we can see that there's a tendency, and the samples down are the ones less heated than the samples above. Actually, we were doing this extraction of the olives at the end of October. So the temperature was still quite high, and we need to put some ice on the bath for the 20 degrees. And then the results of the supervised analysis are here all presented together. What we see is that in the case of the sorry, I just forgot that we were also taking a look to all the principal components, and there was no principal component correlation with the malaxation time. So that's why we decided. Well, actually, we tried to do the PLSDA for the malaxation time, and it was impossible to separate between groups, and that's why we did an OPLSDA, and in this case, it was possible to separate between groups. Here are all presented. What I want to highlight is the case of the PLSDA of the storage, that the 3 and 6 months was almost together, and it was impossible to separate between them, but it was possible to separate between the 0 and the 9 from this group created here. And we hypothesized that this happens because after the 6 months, it started the summer, and then the temperature increased substantially, and then this degradation must be enhanced by this temperature increase. And now let's talk about the marker compounds found, the ones that were giving us information about these differences in composition. And we can see that in the case of the storage, the phenolic alcohols had an increase at the beginning. The phenolic alcohols are mainly tyrosol and hydroxytyrosol that are intermediate products of oxidation. So those increase with small oxidation of the extra virgin olive oil, but then after a while, its composition decreases as its degradation rate it's higher than its production rate. Also, the peroxide and the K270, which are measures of oxidation, increase with storage. Also, the oxidated phenolic compounds, like this one that we have here, that is the oleaceinic acid, the increases with time. Luteolin decreases because it's degraded. And in the case of ripe attribute, this comes from the volatile compounds, and those also have a similar pattern than the phenolic alcohols. The volatile compounds are coming from the oxidation, the partial oxidation of the fatty acids. So during this storage, first they increase its concentration, but then after a time its concentration decrease due to more degradation. Then in the case of the ripening, we saw that when we increase this ripening, when the olives were less green, there's an increase there's a decrease of oleic acid, and there's an increase of linoleic acid. Actually, in the olives, the linoleic acid is formed due to the oleate desaturase from the oleic acid. So this makes sense, as there's a decrease, there's activity of this enzyme, and then the oleic acid decreases and the linoleic acid increases. Also, we saw an increase in the C6 volatile compounds and a decrease in the C5 volatile compounds. As you can see here, those compounds come from the LOX pathway, but the C6 volatile compounds are formed by an enzymatic pathway, while the C5 volatile compounds come from a radical reaction. So it might be that during this ripening process, there's an increase in the activity of this HPL enzymatic activity, and then there's more formation of C6 volatile compounds in comparison to the formation of C5 volatile compounds. Also, in the case of malaxation temperature, there are mainly differences in the volatile compounds. In this case, there's an increase in C5 volatile compounds, and there's a decrease in C6 volatile compounds. It could be because as we increase the temperature, this radical reaction could occur faster. So there's there's increased this part of the pathway than the enzymatic pathway. And finally, in the case of the malaxation time, we see an increase in the apigenin, and there's a decrease in the C6 volatiles. In the case of the volatile compounds, it could be by evaporation or degradation during this malaxation process. And in the case of apigenin, the apigenin is found in the olives in the form of apigenin glucoside. And during this malaxation process, the beta-glucosidase could act and form apigenin. So if you have more time this reaction occurring, then you will have more of this product. So thank you very much for listening to me. And as Rosa said, please, if you want, follow us in the media. We have the Twitter, now called X, Polyphenol Research, and also in LinkedIn. We are very active, and we are always explaining what we are doing, and the new projects, the results that we are publishing, and everything. Thank you very much.
Thank you, Julian. So if we have questions, please feel free to do it. So I think we don't have questions for now. I have one question, two indeed. One thing is, what would you prefer if you have to choose between untargeted or targeted metabolomic analysis?
Well, actually, that's the next step, now that we have all these samples, is to do the untargeted analysis and to compare them. Because, I mean, untargeted analysis, it's like, let's say, more difficult to interpret the results, but it's way, way, way easier to get those results, actually. In this case, we need to send, because we don't have the apparatus for doing all the volatile compounds, so we needed to send the samples to Italy, to send them freeze and everything, and all the cost that it implies. Also, we needed to send the samples to the Agrotechnology Institute, that it's, let's say, kind of near. It's not like sending them to Italy, but it's 100 kilometers from the university, and you have to pay for it and then wait for the results. And in the case of the untargeted analysis, it's just doing some dilution in the case of extra virgin olive oil and just putting them in the machine. And I mean, it even injects like automatically. So you just need them to sit in front of the computer. But as Rosa said, they give different results. So if I have to do just one, I would do the untargeted analysis. But if it's possible, it's always better to do both, I guess.
Okay, thank you, Julian. Also, considering the conditions that you studied for the olive oil, all the parameters, malaxation, temperature, time, and ripening index, what do you think? What was the parameter that most affects the metabolomic profiling of olive oil in general?
The storage, actually, it was in the PCA. The principal component one that was representing it was correlated with the storage. It represented a 26%, and then the second was the ripening index. That it was also I don't remember now, but it was like 12% or something like this. So it's nothing that, I mean, the storage, it's gonna be storage. There's just one harvest a year, so it's gonna be storage always. So you can actually, in those two cases, that you can modify the harvest date, but not the storage. Well, the storage, you can modify the condition of the storage, but it's really expensive to storage at low temperature or freeze or anything.
Yeah.
But
yeah, I agree. Thank you. So, there is any more questions?
actually, there was a question from the before.
Oh, yes, it was a question before. It was if polyphenols undergo sulfatation and glucuronidation in liver to be eliminated in urine, so how would be the human benefit for polyphenols? I think it is. I think it was for Rosa.
Yes, but I can answer if you want. I mean, even if there's sulfatation, those compounds are still bioactive, and also they are eliminated through the liver, but they are like 24 hours in the body, so there's still time of this. And that's why Rosa said about the processing, that there was, if you put the oil, there was more time remaining those polyphenols in the body, so you could have them more time acting inside the body. And we eat all day, like, I mean, during all day you are eating, so you are always having new phenolic compounds inside.
And I would like to add that, for example, there are not many compounds that have health claim in Europe, no, except vitamins and minerals, and only the ones that now have a health claim are polyphenols from olive oil and from cocoa, no, because this means that these compounds, but there are many studies and they have shown a benefit, no, and they are metabolized in the body. I agree that if they are metabolized and they are metabolized to be more hydrosoluble, are quickly eliminated, but they have effect in any case in the body, no?
Yes, thank you, Rosa. Thank you, Julian. So I don't know if there is any more questions. The participants can also email EMN and we will get the questions directed to our speakers. Also, I don't know if Domenica wants to say anything.
No, I'm actually okay, and I just wanted to thanks again the speakers. Great talk. Really, thank you, and thank you also to all the participants who joined us today.
Thank you. Thank you for the invitation.
Yes, thank you.
Thank you to all. So I would like to express my special thanks to our speakers, Professor Rosa and Dr. Julian, and who kindly accepted our invitation to be here in this webinar. And also I would like to let the participants know that the recording of this webinar will be available online if anyone wants to listen to it in the future. And thank you to all the participants for being here, and I hope you have enjoyed it. And thank you all for your attention. Thank you, Rosa.
Thank you. Bye.
Bye. Thank you.
Bye. See you.
See you.
Thank you.